Welcome to Veterinary Physiology lecture. Myself Simpson Soren. Today we will learn the formation of gamete. Gamete means it may be a oocyte or the spermatozoa. So how the oocyte and spermatozoas are formed. So here we will discuss three important points to understand the gametogenesis. First, migration of primordial germ cell, then meiosis 1 and meiosis 2. So let us discuss one by one. The migration of the primordial germ cell. This migration of the primordial germ cell have to migrate to the genital ridge, and before migration this primordial germ cell have to develop extraembryonically. So what do I mean by extraembryonically? After fertilization, when the zygote undergoes cell division or they form the blastocyst, and this when they form the blastocyst, and then it will give rise to epiblast and hypoblast, and during that time some of the cell isolate themselves and they form the primordial germ cell, and this primordial germ cell developed extraembryonically because this epiblast and hypoblast give rise to the three germ layer, that is ectoderm, mesoderm and endoderm, and these three germ layer give rise to the whole organism. So this cell which is isolated, they develop extraembryonically, and when the organisms is developed, they form the genital ridge. This primordial germ cell migrate to the genital ridge. Genital ridge is the primitive gonad. It may be ovary in case of female or testis in male. So when the primordial germ cell migrate to the genital ridge, it will multiply by mitosis division and increase their number. So that is important for the migration of the primordial germ cell, and this primordial germ cell give rise to the oocyte in female and spermatozoa in male. So let us understand the meiosis 1. First we will understand the oogenesis. Here in this lecture we will discuss the oogenesis, and if we understand oogenesis we can understand spermatogenesis very easily. So when the primordial germ cell migrate to the ovary, increase their number, now these cells are called oogonia. Oogonia is the stem cell of the ovary, and when this oogonia undergo meiosis division, that is reduction division, they are committed to undergo reduction division, that is meiosis division. Here the chromosomal numbers are diploid number, and chromosomal numbers are counted by centromere. This red color is the centromere, it is counted by the centromere. So when they are committed to undergo meiosis division, this primordial germ cell or oogonia, they enter into meiosis 1, that is reduction division. They entered into meiosis 1 but they arrested at prophase 1, and that time the oocyte is called dictyate oocyte. They don't complete the meiosis 1 divisions. So oogonia, they are committed to undergo reduction division and they develop themselves to undergo meiosis 1 division. Now this stage it is called primary oocyte. When the primary oocyte, they undergo meiosis 1 division, but they don't complete meiosis 1 division, but they arrested at prophase 1, and now the oocyte is called dictyate oocyte. This is the arrested stage of the oocyte. So all the oogonia, they converted to primary oocyte, they entered into first meiosis division but arrested at prophase 1. So this may be after birth or just after birth or before birth. Many of the mammals, it may occur before birth or just after birth. So at the time of birth in the ovary there is no oogonia. All the oogonia are converted into primary oocyte, arrested at prophase 1. So this is the prophase 1, as this is the diplotin stage where the oocyte is arrested, prophase 1, and now this stage of the oocyte is called dictyate stage. So this is the stage where the oocyte is arrested, and at the birth in the ovary all the oocytes are at dictyate stage. So that is meiosis 1. So to begin with meiosis 2, first the primary oocyte have to complete its first meiosis division. So in first meiosis division we have seen the chromosome number, it become double structure. Even though still the primary oocyte arrested at prophase 1, they are in diploid number but in double structure. So to begin meiosis 2, they have to complete first the meiosis 1 division. So at the time of puberty, some of the primary oocytes are recruited by the ovary, and those primary oocytes are complete their first meiosis division. So not all the oocytes at the time of puberty, few primary oocytes are selected by the ovary and they complete their first meiosis division, and they entered into second meiosis division. And this phenomenon occur in every stress cycle or every menstrual cycle. So every cycle, the ovary recruits some of the primary oocyte to complete their first meiosis division. And when they complete their first meiosis division, it is a reduction division. Now the chromosome number become haploid, but still it is a double structured chromosome. So after completing of the first meiosis division, they form the secondary oocyte and polar body because the cytoplasm content is very less and it is in smaller in size. But another one, secondary oocyte, it is in large in size and more cytoplasmic content. So after the completion of the first meiosis division, they entered into second meiosis division, but again they arrested at metaphase 2. So at the time of ovulation, the ovary released the secondary oocyte arrested at metaphase 2 in most of the mammal except mare and beech. They release the primary oocyte. So at the time of ovulation, most of the mammals they release the secondary oocyte arrested at metaphase 2. So when they complete the second meiosis division, in the presence of spermatozoa, after ovulation, the oocyte will be in the oviduct, and if the animal receive the male or the copulation occurs, when there is semen insemination occurs, this spermatozoa travel to the oviduct, and in the presence of spermatozoa, or when the oocyte is encountered with the spermatozoa, they complete their second meiosis division and give rise to the ovum and another polar body. So this ovum, it is a single structure chromosome. So here secondary oocyte, it was double structure, entered into second meiosis division, arrested at metaphase 2, but after completing of the meiosis second division, now the chromosome is single structure haploid chromosome. So this occurred in the presence of, or when the oocyte encountered with the spermatozoa, they complete their second meiosis division. Without the spermatozoa, this secondary oocyte, they don't complete their second meiosis division. So this is about second meiosis division and the first meiosis division. So here we have learned three important points. First is dictyate oocyte. This is arrested at prophase 1. So all the oogonia, they are converted to primary oocyte and they arrested at prophase 1 at dictyate oocyte at the time of birth or before birth. Second, at the time of ovulation, most of the mammals, they release the secondary oocyte arrested at metaphase 2. So in the presence of, or in with encountering of the spermatozoa, the oocyte complete their second meiosis division. Otherwise, they don't complete their second meiosis division. And the spermatogenesis. What about spermatogenesis? Spermatogenesis, there is no arrested stage. Once it is started, it will continue. So oogenesis begins before birth during the fetal life, but spermatogenesis begins after birth. Once it is started, it will continue and continuously they supply the spermatozoa. So this is about the three important events which we have to remember to understand the gametogenesis. So thank you for watching my videos. If you have any suggestion or queries, you can mail me.